Structural organization in animals. Animal tissues are of four major types: epithelial tissue, connective tissue, muscular tissue, nervous tissue. So epithelial tissue is again classified based on number of layers. Simple squamous epithelium, they have only one layer. Simple cuboidal epithelium, they have only one layer. Simple ciliated epithelium, they have only one layer. You again have the compound epithelium or stratified epithelium. So there we also discussed about pseudo-stratified epithelium. I told you what is the difference between stratified and pseudo-stratified epithelium. Okay. So after that we had discussed types of epithelium based on its shape and function. If they are flake-like cells, we call them as squamous epithelium. They can be simple squamous epithelium or stratified epithelium. The inner blood vessels are lined by simple squamous epithelium. What do we call it as? Endothelium. We call it as endothelium. The body spaces, abdominal cavity and other body spaces, they are also lined by simple squamous epithelium, but there we call it as mesothelium. Remember about endothelium and mesothelium, which is nothing but simple squamous epithelium. If they are found the inner lining of blood vessels, we call it as endothelium. Body spaces, if they are lining it, we call it as mesothelium. Okay. Then I had discussed about based on shape, squamous epithelium, cuboidal epithelium. They are cube shaped. Then we had the columnar epithelium or pillar-like epithelium. So the columnar epithelium or cuboidal epithelium, they might be ciliated also. Ciliated epithelium, where I have shown the cilia. All the epithelial tissue will be having a basement membrane. They are derived from three germ layers: ectoderm, endoderm and mesoderm. I have already discussed it in the previous class. So this ciliated epithelium can be found either in cuboidal epithelium or in columnar epithelium. Now cuboidal epithelium also has brush border, finger-like projections. Brush borders are finger-like projections. The major functions of this microvilli or brush border is to increase the surface area for absorption. Okay. So if you just consider a normal surface, see this is the normal surface. You measure this using a thread. It would be 1 centimeter. Now you measure this finger-like within that 1 centimeter distance itself. If you measure this, it would be more than 1 centimeter. It increases the surface area for absorption, especially in small intestine and large intestine, where the absorption has to take place. You have such microscopic finger-like protrusions. To increase the surface area for absorption, we call it as brush border or microvilli. This can be seen in columnar epithelium or the cuboidal epithelium. Columnar or cuboidal epithelium, you can see this finger-like protrusions, which increases the surface area for absorption. We call it as microvilli or brush border. Now simple squamous epithelium, I have drawn the diagram. They have only one layer and they have flake-like cells with prominent nucleus. So simple squamous epithelium is also called as pavement tissue. They just look like the tiles that are laid on the footpaths. Pavement tissue. Then this is cuboidal epithelium. They might also have cilia or brush border.